Platypus, welcome to Platinum, where future top-notchers are made. I am Andrews, and today we'll explore the functions of membrane proteins. Now, come on and join me as we make our journey to top 1. Did you know that over 60% of every drug in your medicine cabinet is targeting membrane proteins? That's not just a random statistic. Membrane proteins make up about 60% of current drug targets, even though they only represent approximately 20 to 30% of human proteome. But why? It's because membrane proteins are accessible, since they are right on the surface of cells, and they play key roles of controlling what gets in and out of cells, relaying critical signals, and powering essential biochemical reactions. So when a drug needs to block a signal, like in cancer, or help a molecule get into a cell, like insulin, chances are it's interacting with these membrane proteins. Given their central role in both normal physiology and therapeutic intervention, it's essential to understand the various functions these proteins perform within the cell membrane. In today's session, we'll take a closer look at the functions of membrane proteins, specifically transport, receptors, and enzymes. Our learning objectives for this session are as follows. By the end of this lesson, students will be able to first, define membrane proteins and identify their basic structural characteristics in cell membranes. Second, describe 3 of the primary functions of membrane proteins, which include transport, receptor activity, and enzymatic activity. Third, illustrate mechanisms of transport proteins, including channels, carriers, and pumps with specific examples. Fourth, analyze how receptor proteins facilitate cell communication and signal transduction processes. Next is to evaluate the role of membrane-bound enzymes in metabolic pathways and cellular processes. And lastly, compare and contrast the different types of membrane protein functions along with their clinical significance and applications. To start, let's recall what membrane proteins actually are. Membrane proteins are specialized proteins that are either embedded within or associated with the phospholipid bilayer of cell membranes. These proteins can be classified into 2 main categories: integral protein, which are permanently attached to the membrane and often span the entire bilayer, and peripheral protein, which are loosely attached to the membrane surface. The structure of these proteins is unique. Integral membrane proteins typically contain hydrophobic regions that interact with the fatty acid chains of phospholipids, while their hydrophilic regions face the aqueous environments on either side of the membrane. This amphipathic nature allows them to function as bridges between the inside and outside of cells. Given this unique amphipathic structure, membrane proteins are perfectly positioned to control what enters and exits the cell. This brings us to the first major function of membrane proteins, which is transport. Transport proteins are the workhouses of cellular transport, and they can be broadly categorized into 3 types: channel proteins, carrier proteins, and pump proteins. Start with channel proteins. Channel proteins form hydrophilic pores through the membrane, allowing specific ions or molecules to pass through by passive diffusion. Think of them as express lanes on a highway. Once open, traffic flows freely down the concentration gradient. They are very selective. A classic example is the sodium-potassium leak channels. These channels allow sodium and potassium ions to flow down their concentration gradients. The resting potential of most cells is around 70 MV, largely maintained by the higher permeability of the membrane to potassium ions compared to sodium ions. This difference in permeability is due to the presence of more potassium leak channels than sodium leak channels. And another good example is the voltage-gated sodium channels, which are crucial for nerve impulse propagation. When the membrane depolarizes to about -55 MV, these channels open, allowing a massive influx of sodium ions that propagates the action potential. This is why local anesthetics like lidocaine work. They block these channels, preventing nerve signal transmission. Next is the carrier proteins, also known as the carriers, premises, or transporters. undergo conformational changes to move substances across the membrane. But unlike channel proteins, they don't form open pores but rather bind to specific molecules and change shape to transport them. The glucose transporter GLUT1 in red blood cells is a perfect example. It facilitates the movement of glucose down its concentration gradient through facilitated diffusion. The protein has two conformational states, one facing the outside of the cell and one facing the inside. When glucose binds to the outward-facing conformation, the protein changes shape, releasing glucose into the cell's interior. This mechanism is clinically significant because many tissues have different types of glucose transporters. For instance, GLUT4 in muscle and fat tissue is insulin sensitive, which is why insulin helps these tissues take up glucose after a meal. Then we also have the pump proteins. Pump proteins use cellular energy, typically in the form of ATP, to move substances against their concentration gradients. This is active transport, and it's essential for maintaining cellular homeostasis. The star of this category is the sodium-potassium pump. This remarkable protein maintains the concentration gradients of sodium and potassium ions across the cell membrane. For every ATP molecule it consumes, it pumps 3 sodium ions out of the cell and 2 potassium ions into the cell. This 3-2 stoichiometry contributes to the negative charge inside the cell. Why is this so important? Well, this pump is responsible for about 25% of the energy consumption in most cells and up to 70% in nerve cells. It's essential for maintaining cell volume, generating the resting potential, and providing the driving force for secondary active transport. Let me give you a quick comprehension check. If I told you that Digitalis, a heart medication, by inhibiting the sodium-potassium pump, can you predict what might happen to the heart muscle cells? Think about it. The answer is that inhibiting this pump would increase intracellular sodium ion, which would affect the sodium and calcium exchanger, ultimately leading to increased intracellular calcium and stronger heart contractions. Now, let's move on to our second major function, receptor proteins. These are the cellular communication specialists, and they can be defined as proteins that bind to a specific signaling molecule and initiate cellular responses. Receptor proteins are the key initiators of the signal transduction pathway. This cellular process is called signal transduction, also known as cell signaling, and it involves 3 key steps: reception, transduction, and response. First, the receptor recognizes and binds to a specific signaling molecule called a ligand. This binding then triggers a conformational change in the receptor, which initiates the transduction process. Finally, this leads to a cellular response, which might involve changes in gene expression, enzyme activity, or ion channel opening. Receptor proteins come in several varieties, but let's focus on examples that you might encounter in your studies. First, we have ligand-gated ion channels, also called ionotropic receptors. These are dual-function proteins that act as both receptors and channels. When a neurotransmitter binds to them, they open and allow specific ions to flow through. The nicotinic acetylcholine receptor at the neuromuscular junction is a prime example. When acetylcholine binds to this receptor, it opens and allows sodium and potassium to flow, leading to muscle contraction. Second, we have G-protein coupled receptors, or GPCR. These are incredibly important. They're involved in our senses of sight, smell, and taste, and they're targeted by many medications. When a ligand binds to a GPCR, it activates an associated G protein, which then activates or inhibits various enzymes and ion channels. The adrenaline receptor is a classic example. When adrenaline binds to beta-adrenergic receptor, it activates adenylyl cyclase, leading to increased CAMP levels and ultimately to the fight or flight response. Then we also have enzyme-linked receptors, particularly receptor tyrosine kinase. These receptors have enzymatic activity that activated when they bind their ligands. The insulin receptor is a perfect example. When insulin binds, the receptor autophosphorylates and then phosphorylates and other proteins, ultimately leading to glucose uptake and glycogen synthesis. Lastly, our third major function brings us to enzymatic proteins. These membrane-bound enzymes catalyze specific biochemical reactions and are crucial for cellular metabolism and signaling pathways. Membrane-bound enzymes have several advantages over their cytoplasmic counterparts. They can create localized high concentrations of substrates and products. They can be regulated by membrane potential or lipid composition, and they can couple reactions to transport processes. Let's examine an important example. We have ATP synthase, often called the molecular motor of the cell. This remarkable enzyme is embedded in the inner mitochondrial membrane and uses the proton gradient created by the electron transport chain to synthesize ATP. The enzyme has two main parts, the F0 portion, which forms a channel through the membrane, and the F1 portion, which contains the catalytic sites. As protons flow through F0, it causes F1 to rotate, driving ATP synthesis. The process is so efficient that one molecule of ATP synthase can produce about 100 molecules of ATP per second. It's like having a tiny turbine in every mitochondrion powered by the flow of protons. The metabolic significance of membrane enzymes extends far beyond this example. Many enzymes involved in fatty acid synthesis and metabolism are associated with membranes. The enzyme complexes involved in steroid hormone synthesis are embedded in the endoplasmic reticulum and mitochondrial membranes. In fact, the entire process of oxidative phosphorylation depends on membrane-bound enzyme complexes. Complexes 1, 2, 3, and 4 of the electron transport chain are all embedded in the inner mitochondrial membrane, creating the proton gradient that drives ATP synthesis. So now that we've covered the three major functions, let's see how they work together in real physiological processes. Consider what happens when you eat a meal containing glucose. First, glucose in your intestines is absorbed by carrier proteins, SGLT1 and GLUT2, in intestinal epithelial cells. These are our transport proteins in action. Next, the rise in blood glucose is detected by glucose sensors in pancreatic beta cells, which are essentially glucose transporters coupled to metabolic enzymes. This triggers insulin release. The insulin then binds to insulin receptors on muscles and fat cells. Here are our receptor proteins working. This binding activates the receptor tyrosine kinase activity and initiates a cascade that ultimately leads to the translocation of GLUT4 transporters to the cell membrane, allowing glucose uptake. This shows that membrane proteins don't work in isolation but rather as a part of networks that maintain homeostasis as well. Understanding membrane protein functions has enormous clinical implications. As mentioned in our introduction, about 60% of all drugs target membrane proteins. Let's explore some specific examples. In cardiovascular medicine, many drugs target membrane proteins. ACE inhibitors, which is used to treat high blood pressure, target the angiotensin converting enzyme. Beta blockers target beta-adrenergic receptors in the heart. Calcium channel blockers target voltage-gated calcium channels. In psychiatry, many antidepressants work by targeting neurotransmitter transporters. Selective serotonin reuptake inhibitors, or the SSRI, block serotonin transporters, increasing serotonin levels in synapses. Cancer therapy increasingly focuses on membrane proteins. Targeted therapies like trastuzumab or the herceptin target the HER2 or the HER2 receptor in breast cancer, while immune checkpoint inhibitors target immune receptors to enhance the body's ability to fight cancer. Now compare and contrast the different types of membrane protein functions to solidify our understanding. Transport proteins are primarily concerned with moving substances across membranes. They can be passive or active, but their main job is substrate translocation. They are typically highly specific for their substrates and can be regulated by various factors. Receptor proteins are information processors. They detect signals and convert them into cellular responses. They are characterized by their ability to bind specific ligands and undergo conformational changes that initiate signaling cascades. They are crucial for cellular communication and coordination. Enzymatic proteins are catalysts that accelerate biochemical reactions. When membrane-bound, they can take advantage of the unique membrane environment to process both hydrophilic and hydrophobic substrates. They are essential for metabolism and energy production. All three types share some common characteristics. They are all proteins, they are all associated with membranes, and they all can be regulated by various mechanisms. However, ultimately, they differ in their primary functions, structural requirements, and mechanism of actions. That's it! Thank you so much for learning with us today. And always remember, strive, excel, succeed. This is Platinum, where future top notchers are made. See you in the next review.